Hello everyone, my name is Feilicia Gunawan. I'm from grade 12 science and today I will be doing a biology experiment called DNA extraction. Now before we begin our experiment, it's important to wear a bandana or you can use a hair tie to prevent your hair from falling down and disrupting your experiment. Also, do not forget your safety equipment such as the mask and also some pair of gloves. Now for the gloves, you may use rubber or plastic gloves, whichever is more convenient for you. But today I will be using the plastic gloves because they are more accessible to me. So without further ado, let's get started. First, you will need something like this in case there are any spillings or anything, you don't have to clear the entire table. Set it up like this. And then next we will need sodium chloride, or otherwise usually known as table salt. So this will be your table salt. And then next you will need dish soap or liquid detergent. So anything, any brand, you can use any brand, but the one that I have right now is Sunlight. And then next you will need rubbing alcohol. I'm using 70% alcohol, as you can see. And then next you will need a measuring cylinder. I don't have a measuring cylinder, so now I'm using a tumbler from Lock and Lock. And you can see on the side over here there are graduated scales, so I'll be using this. And also you'll be needing 1 teaspoon, and then you will need 2 clear containers, preferably bigger ones. Don't pick something too small. One of them should be big, okay? I'll place them here. And lastly, this is an optional material. This is a food coloring, and I will tell you the reason why I'll be using it. But even without it, it's completely fine. It won't affect your experiment in any way. So now what we're gonna do is prepare our clear container and also prepare about 50 milliliters of room temperature water. And you can measure it using your measuring cylinder or using a tumbler like me. And pour the water into the clear container. Okay, there you go. And then next, we're gonna grab our table salt and our teaspoon. And we're going to place 1 teaspoon of salt into the 50 milliliters of water. Now stir it slowly and gently. Because there are no more salt crystals at the base of the container, so I can proceed. Next, you can grab your other clear container. And you can pour all 50 milliliters of the salt solution into the other container. And what you want to do is that you will gargle the solution for 1 minute. Now while you gargle, you want to prepare your alcohol, your rubbing alcohol, 100 milliliters. So you'll need 100 milliliters of rubbing alcohol. And next, you want to grab your food coloring. Like I said, it's optional. You can skip this part if you don't feel like it. But the reason why I'm using food coloring is so that I'll be able to distinguish between the rubbing alcohol and water. Because they look the same, they have the same color. So I'm a bit worried that I might switch the containers and I might end up drinking the wrong solution. And we don't want that because it will end up really badly. So what we can do right now is grab our food coloring, and we only need a little bit of it, so we just need the tip of the teaspoon like this. And now you can color your rubbing alcohol with this. So now you can distinguish between the rubbing alcohol and the salt solution or the water. So now you don't have to worry about drinking the wrong solution. Okay, now prepare your container. Next, what you want to do is to spit your gargled salt solution, and I have censored it over here because it wasn't pretty. So after you have spit your salt solution that you have gargled, you can pour your rubbing alcohol slowly, just like this. And now you can take your dish soap and you can pour a little bit of it. Okay, just a drizzle of your dish soap. And then use your teaspoon and stir gently to make sure that no bubbles will form. Now we will leave the solution for a while. So as you can see in this time-lapse video, the white clump of DNA moves around the mixture as it interacts with the gargled salt solution. So now after leaving it for 4 minutes, we can see over here, this is the accumulation of DNA. So this white thing that clumps together is the DNA from my cheek cells. For those of you wondering why we had to use salt, dish soap, and alcohol, well, it's because DNA extraction mainly consists of 3 steps. First is cell lysis or cell disruption, second is precipitation, and lastly, purification. In cell lysis or cell disruption, it can be classified into 2 types. One's the physical method and then the other one is the chemical method. When we used the dish soap earlier, it is under chemical method. Why? Because both dish soaps and detergents, they contain a component known as Proteinase K, which dissolves the cellular proteins and therefore can free the DNA from the cell membrane. In precipitation, there are 2 substances responsible. First is the sodium ion, which originates from salt. We all know that sodium ions are positive in nature and therefore they can neutralize negative charges inside the DNA molecules. Therefore, it makes the DNA molecule less hydrophilic, less water soluble, and more stable. Second is the alcohol. Alcohol is a much less polar substance compared to water and therefore DNA is insoluble in alcohol. This causes the DNA to precipitate and be visible to our eyes. Coming to our very last step in DNA extraction, purification. Like its name suggests, this is the very last step to obtain pure DNA by rinsing it with alcohol to remove all unwanted cellular debris. And the colder the alcohol, the faster the DNA will emerge. Congratulations! You have successfully extracted your own DNA, the very basic code of life. You can see the white clump of DNA better at a dark room with a bright light intensity like what I did. Good luck and have fun!